Hello and welcome to this learning session on molecular diagnosis of tuberculosis. In the session 1, you would have heard about various molecular diagnostics which have been used for TB diagnosis. Um, in the session 2, we will be looking at the newer diagnostic tool and the diagnostic tools which are in pipeline. So, I am Dr. Shivakumar from the Department of Bacteriology, ICMR National Institute for Research in Tuberculosis, Chennai. Now we'll look at the newer TB diagnostics which are available or in the pipeline. Um, First among the list is GeneXpert Ultra. GeneXpert Ultra was introduced in 2017 by the company, and this GeneXpert Ultra is different from standard GeneXpert MTB RIF in saying that apart from RPOB gene, there are additional two genes which is responsible for detection of MTB. Now, this with various studies have been proof that it is a non-inferior test to standard GeneXpert MTB RIF, and it predicts both MTB as well as RIF resistance. And in 2017, our WHO also endorsed this test for diagnosis of TB and drug resistant TB. The next test from the same company is GeneXpert Omni, and this is presently in pipeline. And the only difference is that the machine is very portable, and it has inbuilt battery so that it can be used and taken to periphery. And this machine is can be connected with a particular cell phone for downloading the results of a particular patient. So the cartridge used for diagnosis remains the same, and there is no difference in the cartridge. It's the only machine which is made portable compared to the previously available GeneXpert test. The next test has changed both in cartridge as well as the machine. So it is popularly known as GeneXpert MTB XDR, um, and this is presently under pipeline, and it can predict resistance to first line as well as second line drugs. They are isoniazid, fluoroquinolones, amikacin, kanamycin, capreomycin, and ethionamide. The turnaround time is around less than 90 minutes. The processing is same as GeneXpert Ultra or GeneXpert standard MTB RIF. The only difference in the machine is that the previous GeneXpert machine has a 5-color module, whereas if we want to use this cartridge, we need to have a new machine with 10-color module, and the company has also released a 10-color GeneXpert platform. The next test is TrueNat test, which is presently used for diagnosis of tuberculosis in India. This is a chip-based real-time PCR test, and it can be used for both sputum and extrapulmonary samples. There are two devices here. One is the RT-PCR reader and DNA extractor. So DNA is extracted and DNA is loaded onto a chip, and this chip is loaded onto a reader which will carry out the RT-PCR. The advantage of this test is the machine. First, it is lightweight, it is portable, it is battery operated, and can be used as a point of care test at periphery. And the next advantage of this test is that the reagents which are used for this test are quite stable at room temperature, and they are stable for at least 1 year. The total time is close to 1 hour and more, and the preparation time is around 25 minutes and the run time is around 45 minutes. Here you can see that True Prep is the extraction device, True Lab is the reader, and you can see a chip, a green color chip which is called TrueNat chip. Now, apart from the above newly released or tests which are there in pipeline, we can also do in-house PCR methods for diagnosis of tuberculosis. Most popularly used probe is IS610, but in using in-house method, it lacks reproducibility and differ in sensitivity. Also, quality assurance program is an issue when you are using a in-house method. Now we'll come to the next method, which is the most advanced method, whole genome sequencing of Mycobacterium tuberculosis. This method has not yet been endorsed by WHO for diagnosis of drug-resistant tuberculosis, but it can be used for diagnosis of drug-resistant tuberculosis. It will identify the drug resistance by the drug-resistant associated mutations in the clinical isolates, and when you compare the culture, which it gives you drug susceptibility testing, it has the turnaround time is much less compared to the culture, and it has much wide availability or availability of drugs. It Apart from the known drugs, it can identify even novel genetic mutation conferring resistance to existing pool of drugs. So this is the methodology for whole genome sequencing. You extract Mycobacterium tuberculosis genome, you then fragment them, and then subject to the sequencing. Now after subjecting it to sequencing, you perform data analysis in which these fragments are joined together to get a single sequence of Mycobacterium tuberculosis to find out the mutations. So at present, the whole genome sequencing is only possible on culture, though there are studies going on directly to perform on sputum. Quality data relies on meticulous handling of samples and DNA library preparation. Quality control is required for every run of WGS. So data analysis, a skilled bioinformatician is required for data analysis of whole genome sequencing, and at present it is not in regular use for patient management in India. Now, what are the uses of whole genome sequencing? Whole genome sequencing can be used for disease surveillance studies, wherein we can find out how much of drug resistance is present throughout a country, and also we can find out whether there is a drug resistance outbreak in a particular country on a particular region. We can also look at transmission analysis, transmission of one strain to another strain in different groups. It also helps in identifying infection hotspots, which we can use to intensify interventional strategies to control tuberculosis. Now, apart from whole genome sequencing, targeted next generation sequencing is another upcoming field in tuberculosis, which can be used for both prediction of drug resistance. Now, apart from prediction of drug resistance, it can also be used to see whether a person is infected with a single strain or multiple strains of Mycobacterium tuberculosis or non-tuberculosis Mycobacterium. So, this particular next generation sequencing can go up to 15 drugs which are used for treatment of tuberculosis at present, and also can identify mutations for newer drugs like bedaquiline, and repurposed drugs like clofazimine and linezolid. Apart from this, it can identify close to 50 non-tuberculosis Mycobacterium, and it can give you oligotyping results and other genotyping results. So with NT-NGS performed on clinical sample with and on already extracted DNA, average time for comprehensive DST will reduced compared to either whole genome sequencing or to the culture methods. TNGS, if it is used with a portable device called MinION, is a promising DST solution for rapidly providing clinically relevant data to manage complex DRTB cases. So, with this, the key messages include there is a lot of development in molecular diagnosis for tuberculosis, as culture based methods and smear based methods lacks sensitivity, and takes quite a longer time for diagnosis. So molecular diagnostics have played a major role in reducing this time gap, and they are more sensitive compared to the smear and culture based methods. They give quicker turnaround time, and they have significantly enhanced the earlier detection of disease and drug resistance. So the popularly known molecular methods include GeneXpert MTB-RIF presently, and the line probe assay for first line and second line drug susceptibility testing. Now, with so much of advantages, there are some hitches or disadvantages as well, that it cannot detect dead or live bacilli, so it cannot differentiate between dead or live bacilli. Blood tinge sample, if you are using direct samples, cannot be used for molecular diagnostics. um, uh, Mainly, whole genome sequencing cannot be independently used for diagnosis, as it has not been recommended by WHO yet, but it can be used along with other phenotypic methods. So there are possible discordance between phenotypic and genotypic results, which needs an expert opinion. So the point of care devices which are in pipeline are either Omni or whole genome sequencing technologies. With this, we come to end of this session. Thank you for attention.